Hello, my name's Fiona Collins, Dr. Fiona Collins, and our topic today is an update on SARS-CoV-2, COVID-19, and infection control. It's been thought that the first case of COVID-19, the disease caused by the virus, which is SARS-CoV-2, although the titles are sometimes used interchangeably, is a coronavirus. And it was thought the first case occurred late December 2019 in Wuhan, in Hubei province in China. It's now believed that a 55-year-old individual from Hubei province may have been the first person to contract this emerging disease and was confirmed in the first half, or actually November the 17th, 2019. So the virus SARS-CoV-2 itself is a coronavirus. The COVID-19 is believed to be a zoonotic disease, so has its origins from an animal condition hopping over to humans. So it's believed it has its origins in bats, potentially involving bat droppings, and that it may have hopped over to pangolins. And on the right-hand side here, you can see obviously a bat, and below that is a pangolin. And that's a Chinese pangolin, and it's a small anteater. So it's believed that this small reptile then passed it on to humans. It's not the first time that a microorganism has resulted in transmission from an animal to a human. And in fact, other examples include SARS and MERS. And the genome actually of SARS and SARS-CoV-2 are substantially similar. Obviously, we all know that since then, COVID-19 spread through human-to-human transmission with community spread and a rapidly increasing number of cases. The first case in the US was confirmed and reported on January the 22nd this year. Most recently, in fact today, transmission to tigers at the zoo in the Bronx was reported, and one case of transmission to a domestic cat was previously suspected in Belgium. So what is SARS-CoV-2 exactly? It's an enveloped RNA virus. It has a lipid layer. It's hydrophobic, and it actually sticks, and that's kind of a funny term for it. But it has a receptor binding domain that helps it bind to cells. It's got 4 structural proteins. The S, E, and M proteins create the viral envelope, and the N protein holds the RNA genome. The S protein that I just described is the spike protein, and that's what allows the virus to attach to and fuse with the membrane of a host cell. And it actually also helps it gain entry into human cells. Now, I've got a note here about RO and infectivity. So the RO is the reproductive knot or level of infectivity. And it's been stated to be between 1.4 and 2.5. That was in February by the World Health Organization. And that would mean if you took 2.5, that each individual infects, on average, 2.5 people. However, in other studies, the RO has been reported to be between 3.6 and 4, and in another study, as you can see, up to about 3.6. Just as a point of comparison, for the common flu, the RO is 1.3, so 1.3 individuals would be infected by an individual transmitting it, and for SARS, it was an RO of 2. As we know, community spread has been rapid, and the number of confirmed cases has grown. This chart is actually already out of date. This is from yesterday. I did look on the web about 15 minutes ago, and as of then in the US, there were over 364,000 cases. So that was an increase of more than 27,000, and there have been almost 10,800 deaths at this point. So it's obviously everywhere in the world, actually, and I only have some of the countries up here, most of the larger ones. It's a significant concern. The death rate per million varies substantially as well, as you can see. However, it's also known that the actual number of cases may be several times higher. Widespread testing is not being conducted everywhere, and in some places more than others. And on top of that, many individuals have mild symptoms and some individuals have no symptoms. They're completely asymptomatic. So it's difficult to know exactly how many cases there are given those factors. So transmission, I've already mentioned human to human, and it's asymptomatic and symptomatic spread. The main concern has been droplets, so that would be larger particles, spray, splash, spatter, people coughing and people sneezing. However, more recently, there's more of a concern with airborne, and that would be smaller particles that remain in the air for a period of time rather than falling out very rapidly. And that's, if you think about what's just happened with the CDC, there's now a recommendation to wear a mask of some type in public. Fomites are found on surfaces, so if you think about the surfaces, not only in the dental setting, but elsewhere as well, the virus can land on surfaces as well. And there's been some indication of a fecal-oral route as well, with the viral DNA being detected in stools in infected individuals. The other thing is how long does it last? How long does it stay on the surface? So it's estimated that it can remain airborne for 2 to 3 hours. The viability on plastic and steel is considered to be up to 2 days, and cardboard 1 day. I think probably most, if not all of us, are now social distancing, and it's been recommended that that be conducted at a minimum of 6 feet apart. However, there is recent concern regarding those small airborne particles, and there is some research that suggests up to 23 or 24 feet is the reach of those smaller particles. The other concern, of course, in the dental setting is aerosols. I will discuss those shortly. So if we think now about the dental setting, we know offices have closed, and the current recommendations are to postpone elective procedures, surgeries, and all non-urgent dental visits. Obviously, the objective is to keep patients safe, keep staff safe, and also to preserve personal protective equipment and supplies. Another concern is that most dental settings don't have airborne infection isolation rooms. They don't typically stock N95 respirators, and they don't typically have a respiratory protection program. And that all relates to the N95 respirators. Another recommendation is that if a staff member or you are sick, is to stay at home. And the other recommendation is if an individual does call with an urgent need for care, the recommendation is to screen those patients on the phone for COVID-19. If an individual has had no symptoms, no known infection, then they could be treated in the dental setting. If the patient had COVID-19 and at least 3 days since their recovery and at least 7 days since the COVID positive test, they could be treated. In other words, the patient has to have completed their home isolation. However, if the patient, if you have a patient in the dental setting who arrives and has suspected or confirmed COVID-19, you would want to defer non-urgent care, and if treatment is urgently needed, refer that individual to an appropriate facility. You'd also want to clean and disinfect environmental surfaces per the CDC guidelines using an EP-approved emerging viral pathogens claim disinfectant. And we'll talk about that later as well. So if we think about standard precautions, it includes everything on this list. So it would include hand hygiene, PPE, or personal protective equipment, respiratory hygiene, etiquette, sharp safety, safe injection practices, sterile instruments and devices, and environmental surfaces management. All of those are key components of the protocols in the CDC recommendations for the dental setting. Now, we just talked about offices being closed, and we talked about under what circumstances to treat patients. Standard precautions obviously apply still. However, while in some of these protocols nothing has changed, in others it has, and there's always the need for vigilance and making sure to follow procedures and protocols in general. And I think one of the things this pandemic has done is really highlight infection control. It's really, I believe, uppermost in most people's minds, including the general public, in terms of how they can avoid transmission and passing it on to loved ones and friends. So transmission-based precautions are a step up from the precautions typically required in the healthcare setting. And they step in situations where there's an epidemic or a pandemic. Let's turn now and look at hand hygiene. There have been several questions about hand hygiene. Do we need to do anything different? What should we use? So there are several things really to be considered, and these are considered generally for standard precautions as well. So one is, are your hands visibly soiled? If your hands are visibly soiled, using an alcohol-based hand rub will be ineffective because that cannot remove soil. So if hands are visibly soiled, it has to be soap and water. We're talking about routine procedures. That soap can be either plain soap or antimicrobial soap. If you then would thoroughly wash your hands for at least 20 seconds, you would do that at the beginning of the day before donning gloves. Obviously, the beginning of the day may be one patient at the moment, on not many days, it depends. But regardless, before donning and after doffing gloves, if there's any contact with the patient, ungloved contact, if there's contact with a potentially contaminated surface, and obviously at the end before you leave the operatory. So those are examples of when to perform routine hand hygiene. You'd want to consider whether an individual has sensitive skin and allergies. The best barrier actually for hands is the skin. It's a huge organ and it is a great barrier provided it is intact. If you have somebody with sensitive skin and allergies, or if you don't dry your hands very thoroughly, or you use water that's too hot, you can end up with dermatitis, irritation, and broken skin. So you've basically lost that barrier. Prior to washing hands to perform hand hygiene, you would remove all jewelry and scrub nails. The nails should be kept short because otherwise there's a risk of contamination and transmission of microorganisms because you can't get under the whole of the subungual layer because the nails are so long. Too much washing can also cause skin to crack. The other thing that's important, given that skin is such a good barrier, is to use a water-based moisturizer to help keep skin intact. Hand washing provides mechanical removal of the soil. It's also important to dry your hands thoroughly. Hands that are left damp also become irritated and can also cause irritation and broken skin. For surgical hand hygiene procedures, you would still need to follow the hand hygiene recommendations for surgical procedures. Hand hygiene sanitizers or alcohol-based hand rubs are actually the preferred method per the current CDC recommendations. They're considered to be the primary method provided there's no soil. The recommendation is to use an alcohol-based hand rub with more than 60% ethanol and more than 70% isopropanol, either or. Hand hygiene or alcohol-based hand rubs containing emollients also help to protect the skin and prevent irritation. So these alcohol-based hand rubs effectively reduce the number of pathogens that may be present on the hands and also will avoid frequent hand washing. By avoiding that, you're potentially having less damage to the skin. Many people think that alcohol-based hand rubs are actually more irritating than soap and water. In fact, the converse is true. They are less irritating. You're not using water. You haven't got wet hands. You don't have to dry your hands. You're not relying on a specific temperature range for the water to reduce irritation, and they contain emollients. So for all of those reasons, they actually help to preserve the moisture in the skin and are less irritating. They're easier to use, which is good for access and can help with overall compliance. And in fact, they also have been shown to be more effective, provided again, there's no soil. One of the questions that's been coming up is whether you can use a benzalkonium soap or benzalkonium chloride. That's actually got less reliable activity, and the CDC is not recommending that during the current coronavirus epidemic. Personal protective equipment typically includes gloves, masks, protective eyewear or face shields, protective attire. Now we can see with gloves, we've got two different types here. The upper one is a routine medical glove. So it's a single-use disposable glove used for routine patient care. If it's a surgical procedure, which is actually more likely given that it's emergencies that are being seen at the moment, then you would use sterile surgical single-use gloves. The ones on the lower right are the heavy-duty utility gloves that are indicated for operatory cleanup and for instrument reprocessing. If you look here now at the masks and the respirators, we can really see the differences between ASTM levels 1, 2, and 3 and NIOSH 95, so N95 respirators. There's greater filtration efficacy. The fluid resistance is the same. It's still the same flame resistance, but significantly greater filtration efficacy as you go from an ASTM1 up to an N95 respirator. So there's greater filtration efficacy. One of the concerns is airborne infection and aerosols, which we touched on a little while ago. The two largest causes of aerosolization in the dental setting is during procedures using an ultrasonic scaler or a high-speed handpiece. Now, if you think about the current situation, ultrasonic scaling is not an emergency. It would fall under elective, non-emergency. So aerosolation with an ultrasonic cleaner should therefore not be a current concern. We do need to consider high-speed handpieces. Using a rubber dam whenever possible to treat an emergency patient and a high-volume suction both help to reduce aerosolization. In fact, I was talking to a dentist last week, and he was saying that he makes sure to use a rubber dam even for extractions if he's sectioning the tooth. And of course, I'm sure it makes it more difficult. I've not done that with the rubber dam in place, but it certainly makes sense in terms of reducing aerosolization. The other thing is that the American Dental Association currently recommends preemptive rinsing with a 1.5% hydrogen peroxide solution or a commercially available rinse that contains 1.5% hydrogen peroxide. So that would be used just prior to beginning treatment. It is an off-label use if you're using a commercially available rinse. And there are studies showing reductions in microbial load. It's not known if it specifically reduces coronavirus levels, but as a general precaution, it reduces microbial load and is recommended. We then look at the N95 respirators. They're significantly more effective than masks, as I mentioned. They're higher filtration rate. And the other thing is, if you think about how you wear a mask, and I'll just pop back a slide for a second. You can see here that this young lady's modeling. She's putting on the face mask, showing the lips going behind the ear, then stretching it out below the chin, pinching the nose band of metal so that it sits nicely over the nose with a tight fit. But at the sides, there's usually a small gap, and microbes, as well as other substances, can enter through those gaps. So they're not as secure, and the seal is not as good as with an N95 respirator. In fact, you can't call it a seal, it's covering the area. So the N95s do provide a seal, provided they fit well and they've been fit tested. Facial hair around the seal area is a problem. It will not be able to seal. So the recommendation there would be to remove facial hair around the area where the respirator would seal. You can see 3 different types here. These are all examples of N95 respirators. One of the questions that come up is, well, how do I know this is a NIOSH or an N95? There have been some reports of counterfeit products lately. And so this diagram actually shows that you'll see a model number, you'll see a lot number. The lot number is recommended, model number is required. But you also, on the left-hand side as we're looking at it, so the right-hand side of the exterior, can see the name NIOSH in block letters. There's also a filter designation with the rating as well, and the filter efficacy. So those are ways of identifying an N95. Hopefully the counterfeit product doesn't have that, but you can also look up the model number. I've mentioned it requires fit testing. So initial fit testing is required before using a respirator. It's required when there are changes in an employee's physical appearance, so or conditions. So for instance, if you've lost a lot of weight, that initial fit test may have confirmed sealing at the time around the face, but it may no longer apply if the person's lost a substantial amount of weight. It also needs to be performed when a worker starts to wear a new model or type. If it doesn't fit, it doesn't form a seal, and then some of the air will bypass the respirator's filter. There's usually a requirement for an annual fit test. That has been temporarily suspended, but the initial fit test is still required. One way to do this that doesn't destroy an N95 is to do a qualitative fit test. And that's a pass-fail, and there is actually one that is indicated and recommended. It relies on the user tasting or smelling a substance. And it's a test agent that can be saccharin, it's a sweetener, obviously, or Bitrex solution, which is bitter. And this qualitative is the preferred method. It does rely on the wearer being able to report a taste if there's a lack of seal for the N95 respirator. If they can't taste because their taste is poor, they may say they don't taste anything, but it doesn't necessarily mean the fit's great. There are kits that are available, should you be interested, with suppliers providing saccharin or Bitrex. There is an additional screen that can be performed each time. This doesn't obviate the need for fit testing, but it's a quick and easy check that might give you peace of mind if you're using your N95. And you can basically place your fingers next to the seal, exhale forcefully, and see if you can feel any air on your fingers reaching through from the inside of the respirator to the outside. If there is, it means that there isn't a good seal. So you could place a finger at the 12 o'clock position, the 6 o'clock, at the 3 o'clock and the 9 o'clock. And then after that, you could also test the 11 o'clock, 5 o'clock positions together, as well as the 1 o'clock and the 7 o'clock positions. We're still hearing about N95 shortages, and in critical shortages, they are usually single-use disposable. Let me start with that. But in a critical shortage, in the current situation, an N95 respirator, it is acceptable to reuse it, provided it's not soiled. That reuse must be by the same individual who used it before. There's also the possibility of reducing the risk of soil. One option is that's been suggested is to wear a face mask over the N95 respirator, so that if any soiling does occur, it would go on the face mask and hopefully not reach through to the N95 respirator. Another method which really helps is to use a face mask, or a face shield rather, over the respirator, and use that instead of other protective eyewear. So if we look at protective eyewear, often that will be some protective eyewear with side shields, and it will fit over the eyes, above the brow, below the eyes, and seal in those areas. Those are great for protecting ocular mucosa and the eyes. However, if you use a face shield, you're also going to have a layer of plastic basically over a large part of the face, which will protect the skin as well. And as I mentioned, it will also help to protect those N95 respirators. One of the other questions that came up before I move on to clinical contact surfaces is whether staff at the front desk should be wearing PPE. So this question came up in relation to COVID-19 and the risk of transmission. I think the question really is, since only emergencies are being dealt with, do you really have any need for staff at the front desk? Can you manage without those staff? If you can, it helps to reduce potential exposure, therefore reducing the risk of transmission. It also alleviates the use of PPE when supplies are short. So bottom line, the best is probably to first assess, do I need a front staff person? Do I need anybody? Do I need a staff member at the front desk? Can I manage on my own with an assistant, or can I manage on my own? So now let's look at the clinical contact surfaces. We talked earlier about SARS-CoV-2 being a lipid medium-sized virus. Now that actually is a piece of good news, if there is any. And it basically means it's one of the least resistant microorganisms to disinfect. And as you look at this chart, you can see lipid or medium-sized viruses are bottom of this totem pole. So they're actually the least resistant. Obviously, bacteria and others are progressively more resistant the further up this list you go. The procedure itself for cleaning and disinfecting clinical contact surfaces really hasn't changed. The procedure is no different to before. You have to clean to remove soil, and that's followed by disinfecting with an EPA-registered hospital-level disinfectant. Now, if you've typically used a one-step product, it's still essential that visible soils first removed if present. Under the current pandemic, the CDC recommendation is to use a hospital-level disinfectant, and the EPA has provided a list N, which contains the products with emerging viral pathogens and human coronavirus claims, and it's recommended that one of those be used during the current pandemic. It's a very long list. I first saw a list on March the 17th, and it was updated again, and the latest update was April the 2nd. So it is being periodically updated as more products meet the requirements for this. You'll actually find many products on the list, including brands we're familiar with. It's a very long list of products. So examples of ones we would be familiar with that are on this list include Cavicide, Clorox Healthcare, VersaSure wipes, SaniCloth germicidal wipes, PDI SaniCloth, and Decide Ultra. Regardless of which product you're using, as usual, it must remain wet on the surface for the recommended contact time. That varies greatly. There are some on the list that's 30 seconds, up to 5 minutes, and in fact, there are some that I didn't actually recognize the names of that are 10 minutes. I haven't seen those in the dental setting, but it does vary. It must remain wet on the surface for that recommended contact time. One question that typically comes up is, or sometimes comes up, is that the wipe isn't keeping it wet. There's not enough liquid in the wipe. Now, that could happen if the canister was slightly open. Some of them, as you know, have got a two-click closure. If you clicked once and it didn't actually get fully closed, there would be some evaporation of the liquid. That could cause there to be less liquid in the wipe, which means there isn't the required amount in the wipe to coat the surface properly and leave it wet for the contact time. Another reason is perhaps the wipe is being used for too large an area. If you don't know how long the wipe needs to, or what area it can cover, if you look on the labeling, it will tell you in the IFUs what square area the wipe is suitable for. If you do that, then that will help to keep the surface dry, wet for the contact time. Talking of drying, some people have said, well, sometimes if we're in a hurry, we'll wipe and then we'll use a towel to dry off after a minute. Now, If you have a 2-minute contact time, that means you haven't met the contact time, which is the kill time for the microorganism that requires 2 minutes. If you want to find out if the product you're using is on the list, you could take the first 2 sets of the EPA registration number, go onto the EPA website and do a search. You could also download the list N from the EPA website and then do a search that way. I think it's about 25 pages long. It's a very long list, but that avoids you having to go through the whole list. The question that pops up is, well, how long does it actually last on the surface? So based on current information, as I've mentioned earlier, it's on plastic and stainless steel up to 3 days and up to 1 day on cardboard and about 3 hours in the air before it falls out of the air. Clinical contact surfaces can also be handled, of course, using FDA-cleared barrier protection, and that's still the case. The caveat is when you remove it after use for a single patient, if there's visible soil underneath when you've removed it, then you still have to clean and disinfect that surface. Let's now look at instrument reprocessing. Really, not a lot has changed. It remains a key activity in infection control, as before for critical and semi-critical heat-resistant devices and instruments, as well as handpieces and other attachments. Heat sterilization is recommended and meets best practices as long as it's properly performed. Now, you would need to follow the recommended protocols, have appropriate reprocessing equipment that's fully functioning, keep the required logs and documentation. And of course, the focus is on safety, effectiveness, and usually efficiency as well. There are a series of steps, sequential steps that are involved. And we can see at the top, we're disposing of single-use items first, appropriately. And then I've got POUs, that would be point-of-use cleaning. That may or may not be recommended in the instructions for use for a specific instrument. If it is, point-of-use cleaning should be performed as indicated in the instructions for use from the device manufacturer. That will remove gross debris immediately after patient care, which helps to prevent it from drying on the exterior prior to its transportation to the reprocessing area or room. From the previous slide, we saw it was a young lady, actually a student, and I'll just flip back for a second. We can see here there's actually an instrument cassette or an IMS system cassette inside this tub. So these are used instruments and a contaminated cassette. And while instrument cassettes are much safer than taking instruments loosely through to the reprocessing area, it's not impossible for an instrument with a sharp point to poke through the perforations. So that's the rationale for placing them in a tub with a closed lid. So it needs to be puncture resistant, rigid, and with a closed lid. And that meets OSHA requirements for helping to prevent risk of exposure to bloodborne pathogens. You'll also notice that heavy-duty utility gloves are being worn. So this was operatory cleanup initially, taking it through to the reprocessing area. And in fact, reprocessing starts with point-of-use cleaning. So from then on, it should be heavy-duty utility gloves. We then look in the following stages are cleaning. There is another potential stage once you've transported them into the receiving area. So in the receiving area, you would segregate the instruments. And then if there are some that can be pre-soaked, they should be pre-soaked under 2 conditions. This could happen to prevent debris from drying on if the instruments won't be further processed for some time. It's also helpful if debris had already dried on, and it helps to moisten it so that it's easier to remove when cleaning does occur. Again, this should only be performed for instruments and devices where the instructions for use indicate that pre-soaking is advisable and is an option. The reprocessing area has to have a unidirectional workflow. So when everything arrives, when you take it through from transportation to the receiving area, that's obviously the dirty end or the contaminated end. It would then flow through each of these stages. So the instruments would then move along the line within the room from dirty to clean, being cleaned, then inspected for damage and to check they're clean, for packaging and labeling, for sterilization, after which they are sterile, provided they are intact packaging that was not taken out of the sterilizer before it was dry. The other aspect is monitoring and logging sterilization processes, and that's crucial because frankly, otherwise you don't know that you have produced sterile instruments at the end. They may still be contaminated. And the last stage, of course, is storage in a dry, dust-free area. So let's look at cleaning. We talked earlier about alcohol-based hand rubs not being able to remove soil. Well, if you leave soil on instruments, the sterilant, typically steam in our case, in autoclaves, cannot penetrate the soil. So it compromises the ability to sterilize those instruments. So cleaning is an absolutely critical step. You'd want to look at the design and features of equipment when you're choosing it. Which equipment is appropriate for which task? Again, how efficient is it? How long does it take to perform the process? How effective is it in performing the process? And does it contribute to worker safety? The last one, of course, is you certainly want it to have a long shelf life. So for cleaning, I only have 3 options up here: instrument washers, instrument washer disinfectors, and ultrasonic cleaners. We all know that manual cleaning exists. It is not recommended and it's discouraged unless required for spot cleaning or for a specific area of a specific device. So it should be avoided when possible, and automated instrument washers or washer disinfectors or semi-automated ultrasonic cleaners are recommended. If you're using an instrument washer or a washer disinfector, that is fully automated. You can place instruments in there, you can place cassettes containing contaminated instruments in the instrument washer or washer disinfector, and it will run through the whole cycle. It will provide a pre-wash. Cold water will remove the dirt or the soil. There will be a wash with detergents, so that will provide for cleaning. There will then be a rinse. And in the case of the instrument washer disinfector, there will then also be a thermal disinfection cycle. So that uses high temperature water above 90 degrees Celsius. And that actually helps to reduce the microbial load before you go to the next stage. For both types, washers and washer disinfectors, hot air drying occurs in the device. So it means when you take it out, the device has been rinsed, it's been cleaned, it's been rinsed again, and it's been dried. So there's no requirement to dry it. In the case again of the washer disinfector, you've also reduced the microbial load. In the case of ultrasonic cleaners, you would first of all place them in a rack or a designated container, and then place them in the ultrasonic cleaning solution. It's important that you don't place them on the base of the chamber or at the side, against the sides of the chamber. The way the ultrasonic cleaner works, obviously, is it cavitates, it produces bubbles. Those implode, release energy, and that cleans or mechanically cleans debris from the instruments that are in the solution. So if you have them against the base or against the sides, you can't get proper cavitation because there isn't space there between the instrument and the ultrasonic chamber itself. So you won't get implosion in that area, therefore it won't be clean in that area. So if you're using cassettes, you would place them in a rack. We were talking about aerosolization. It's important before starting the ultrasonic cleaner that the lid be closed, and that prevents aerosolization of bacteria. It's also important to leave that lid on until the ultrasonic cleaner has stopped. There are also cleaning efficacy tests available now for instrument washers, instrument washer disinfectors, and ultrasonic cleaners. The next stage is inspection. So you would inspect the instruments to make sure they're not damaged and they're free of any debris and soil. After that, you would move to sterilization packaging. Again, they have to be clean, and the instruments and cassettes, if you're using cassettes, also have to be dry. The sterilization packaging is FDA cleared. It needs to be strong and tear resistant, and it has to allow penetration of the sterilant. If it's been FDA cleared as packaging, it's been tested for penetration, and there is data that's provided on penetration of the sterilant and the strength. The other thing that sterilization packaging does is it verifies your sterilization parameters by using chemical indicators. So the parameters are time, temperature, and pressure. The critical ones are time and temperature. Pressure is the third one. So those parameters, the chemical indicators, will let you know if it reached that parameter. Now, since the chemical indicators change physically, it also avoids confusion between dirty and clean instruments that are because they're within this packaging with the chemical indicators. The instruments will remain sterile in the packaging as long as the packaging is intact and until it's opened. Immediate use steam sterilization, which we all also know as flash sterilization, does not meet best practices. Best practices and recommendations are to use packaging. Immediate use steam sterilization in the dental setting is permissible with several caveats. It needs to be performed as close as possible to where it will be used. It needs to be taken to be used immediately after it exits the sterilizer. And if you think about it, that's logical, because as soon as it comes out of the sterilizer, it's subject to contamination because it is not enveloped in intact packaging. It's also not to be performed for routine procedure. It's for an exceptional circumstance. There's one exception, and there are closed cassette chamber sterilization devices in the dental setting. Those do not need sterilization packaging for the devices being processed because it is a completely closed cassette chamber. However, if you're taking it out of the closed cassette because you want to use the closed cassette for another purpose, rather than because you want to use the device, then at that point the device is subject to contamination. So the caveat really is it staying in that closed cassette chamber? I'm not going to go through this table, but as you can see, several methods of sterilization. Gravity displacement, as the name suggests, the air is displaced by gravity at the bottom of the chamber. The others all involve pre-vac cycles, and the pre and the post-vacuum obviously includes a post-vacuum cycle, which helps with post-vacuum, it provides post-vacuum drying. The times vary. You have to add on the drying times, and it depends on the device, it depends on the type of device, and it depends on the temperature that was used. We now look more closely at sterilization monitoring. Mechanical indicators, chemical indicators, which I'd already mentioned, and biological indicators are the three types of sterilization monitoring that we should use. The mechanical indicators used to all be analog. They're mostly digital now, and you can see a couple of examples here. One on the left is a digital, and you can see the cycle parameters are here. They've got the temperature. Here we've got the chamber temperature, we've got the chamber pressure as well, and we've got time too. So those are the parameters. It doesn't say how long it was there necessarily. So you need to look at the mechanical indicator, whether it's digital or analog. The digital are certainly more sophisticated, and you can get printouts of them or load them onto a USB port and then archive them that way. The chemical indicators, as I've mentioned, confirm that you reached, or the sterilizer rather, reached the parameters. It does not provide sterility assurance in any way, shape, or form. It does help confirm, or it does confirm that it reached the parameters, that's all. The biological indicators provide sterility assurance. So biological indicators are only a biological indicator with resistant spores can measure the microbial killing power. And that's because the spores are at the top of the totem pole, if you like, for resistance of these microorganisms. So if the spores have been killed, the less resistant microorganisms have also been killed. The CDC recommendation is to perform that at least weekly, and it should be preferably performed daily, at least daily. The other aspect is every time an implantable device has been sterilized, a biological indicator has to be included. In our setting, implantable device here does not mean a dental implant. Those are sterile, typically in a double vial and gamma irradiated before they arrive in the office. In fact, if you were to place them in an autoclave, if it's in the vial, it won't do any good. And in fact, the implants can actually be damaged by autoclaving. So it's not referring to implantable devices. So anyway, preferably daily, certainly at least weekly. If you think about it, daily use provides more frequent assurance, which also gives peace of mind. And I would expect, particularly given the current situation, we'd want to know for the load that in fact sterility was assured. Biological indicator is also best practices and standards. Now, why would it fail? Inherently, we probably want to think it's the spore test that failed. But in fact, many studies have shown that operator error is a major cause of failure. In one study, it was 87% of failures were due to operators not operating the equipment correctly. So here are some examples. Manipulating or interrupting a cycle. That could show up in mechanical indicator records. Now that there are automated cycles, for many autoclaves that's not possible anymore. But those of us that were in practice before automated cycles can probably remember that if you wanted to, you could pop the door open partway through a cycle and interrupt it. Frequent operator error causes are incorrect packaging. You use the wrong packaging. It's too thick, it's too much, it's the wrong type. Overloading the sterilizer. And that's really where the accessories that come with sterilizers are so important. So there are racks that will provide the correct amount of space between packaged cassettes and that will allow the sterilant to get in between those spaces and penetrate the packaging. So that's one example. There are also side-on racks that will allow you to place paper plastic pouches at an angle as well, with sufficient separation between them for the sterilant to get in there. All of these causes can actually prevent contact of the sterilant. Incorrect time, using an incorrect time or an incorrect temperature can also cause issues. Poor maintenance. Are the gaskets damaged? Do you need to replace the gaskets? Are the seals gone? So there are actually, there are others as well, but these are frequent causes of operator error. So now let's look specifically at the biological indicator options. One option is an in-office method, and the other is a mail-in. Some people like using a mail-in because it provides third-party verification. However, an in-office provides you with much quicker results. Considerations then are quick testing within an office one for sterility assurance. Again, peace of mind. You don't need to quarantine instruments for as long. So it allows you really to take control of your steam sterilizer testing. It does give you peace of mind. It lets you know there and then, almost there and then, that your sterilizer is functioning properly with more rapid results. In-office biological indicators or monitoring, it does help comply with best practice guidelines established by the CDC. And it states that steam sterilizers, again, they should be monitored at least weekly, preferably daily. They're quick, they're accurate approaches to steam sterilizer biological monitoring, and they've been tested. We can see several here. All of these are user-friendly. They're suitable for gravity and vacuum autoclaves, so you don't need to have different spore tests for different autoclaves if you have more than one type in the office. And so here we have three different ones. The one on the left is called a confirm, and that's Crostex. That's got a 10-hour test, and there's a 24-hour test. The one in the middle is a Tuttenauer product with a 1-hour and a 20-minute test. And the one alongside that to the right is Spore Check. And the Spore Check is a 24-hour test. You can detect results, start to detect results in as little as 2.5 hours. So there's quite a variation in the length of time required for the testing. they are all quicker than mail-in testing. Some now also have, you can also get a printout, which can also be placed in the monitoring log. It's essential that a monitoring log is kept. If you don't keep a monitoring log and there are any issues, it will be assumed that you did not do so. It needs to include the load number, the sterilizer load, the sterilizer number, serial number, particularly if you've more than one. It should contain the contents of the load. It should contain all of the monitoring results. So that would include each time results of mechanical monitoring, chemical monitoring, and biological monitoring whenever it is performed. If you have printouts, they should be kept in the monitoring log. At the end, when you've got everything in that monitoring log that you need, it also needs to be signed and dated by the operator. So recent developments, there have been quite a lot of recent developments. If we start with technologies, we just talked about increasingly rapid spore testing. There's now an option in one series with a preheating stage, and it's a preheating stage of 15 minutes at a minimum temperature of 103 degrees C. This is a new addition. It's actually Tatnar, and during that initial period, there's no air venting during this time, so it reduces microbial load. It's conducted at a sufficiently high temperature that it would kill viruses and also other microorganisms. So it's not rendering anything sterile. You still need to run the sterilization phase, but it does help to reduce microbial load. We're also seeing people optimize N95 respirator supply, and there's been, as we all know, a shortage. I mentioned some guidelines during COVID-19, including reuse by the same operator. Production is reported to be increasing, and interestingly, there are decontamination methods also being developed for N95 respirators in the current crisis. There was vendor approval from Battelle, and that was for a vaporized hydrogen peroxide technology. And this is being used in huge chambers that look like rooms where the N95 respirators are being decontaminated. Again, they have to be not soiled. If they're soiled, you cannot use this technology. It's for decontamination, not for cleaning. There are other methods in development as well, and one of them uses a lower heat temperature, and there are methods that cannot be used. So some of the questions that have come up with respect to masks and respirators have included, can I wash the masks if I'm really short? Well, not only are they single-use disposable devices, but in fact, if you wash the masks and or respirators, it destroys the barrier properties. You can't use steam sterility, that will damage them. High temperature will affect the elastics. If you use a standard sterilization technique, it has also been shown to be able to affect the joint between the metal area that you create a divot in when you put it on. So really, there weren't really any options, and people are stepping up to the plate with these new technologies to help with N95 respirator desperate shortages. More testing options are in development as well, both to detect the infection. So there are more options, there's becoming more availability, and there are quicker options as well. There are also testing options that exist in laboratories and others that have been in development to detect immunoglobulins, M and G, after a person has been infected. And that will detect the level of immunoglobulins and immunity, at least at that moment, for that individual. There are also drugs that are being developed and being investigated. So one antiviral that was developed actually for Ebola that's being looked at and is in late stage trials is remdesivir, and that's an antiviral agent from a company called Gilead out of Israel. There are also other drugs and other methods that are being investigated as well. So, as I mentioned, more availability and faster tests, more testing on the presence of immunoglobulins. So really, we've given a brief overview of certain aspects, background on SARS-CoV-2 and COVID-19. It's clearly a very serious pandemic. We should all be social distancing and doing everything we can to prevent transmission. outside of the dental setting. And there are certainly CDC recommendations, there's FDA guidance, and there's EPA guidance and recommendations currently on what to do during the COVID pandemic, as well as from the ADA. What I've discussed today is current as of now. It's been a rapidly developing situation. And in fact, a week ago, some of the information that's out wasn't out. Certainly the epidemiology is advancing, and methods of detection are advancing as well. So with that said, I'd like to say thank you, and if you have any questions. Okay, so I have a question here about if someone dies and they test positive, the cause of death is automatic, says COVID. I haven't, to be honest, I haven't been involved in that aspect of things, so I don't know if that's always automatic. If it does, it could skew the numbers. However, if the person, well, it's a difficult one. I really don't know the answer in terms of is it automatically says COVID. It's possible if it says some signs of COVID, if it then says that it was listed as the cause. If there was an underlying condition and COVID was the cause of immediate death, then that was certainly a factor. What about asymptomatic carriers coming into the dental office for emergency care? So as this is obviously a problem, we don't know who asymptomatic carriers are unless everybody or most people are being tested, which they currently are not. So that's part of the reason for taking extra precautions for people who are coming in as an emergency. If it's somebody who is symptomatic, I had discussed earlier under what circumstances you could treat them in the dental office with appropriate procedures in place and appropriate PPE. But there were also examples of people who clearly were symptomatic and not past the home isolation period, and they would be referred. So yes, there may be an asymptomatic carrier. The screening is going to weed out people who are actively ill. It's not obviously going to weed out others. Let's see. Can you use expired hand sanitizer? How long can you use it after? The expiration date's the expiration date. So honestly, I don't have any opinion on that. It shouldn't be used beyond the expiration date. So it's not an opinion, it just shouldn't be. Is bleach a good disinfectant? Bleach can be used at home at a dilution of 1 to 10. It's not recommended. The recommendations in the dental setting are the substances that are on the EPA list N. And some of those do contain bleach as one of the ingredients. Will there be an updated PPE requirement once we return to normal operations? Well, I don't have a crystal ball, but I would have to say I think some things could change once we get back to our new normal. But the answer in terms of do I know? No, I don't think we know what will be required afterwards, if there will be an update, but it wouldn't be surprising. Okay, how do you use saccharin to help test N95s? It's provided in test kits, and you actually use a nebulizer, and then the person lets you know if they could taste any. So the nebulizer puts it in the air, and then they're breathing in. So the aspect is if there's a lack of seal in one area of the N95 respirator, that they will breathe that in, and because obviously of the connection between the nose and the mouth, that they will then taste the saccharin. What's the best way to disinfect face shields? You could use soap and water. You could also use a disinfectant wipe. And the caveat is check with the face shield manufacturer that what you want to use is compatible with that face shield. Okay, so the question is Cavicide considered a low-level disinfectant? Will it be suitable for COVID? So Cavicide is an intermediate level disinfectant, and it is on the list N. If you go to the EPA list N, it and some other disinfectants that were used in the dental setting that I'd mentioned are on that list. How long are instruments recommended to be in a cold sterile before bagging and placing a sterilizer? They're not recommended. They should not be placed in a cold sterile solution. I'm presuming this is the FDA cleared high-level disinfectant. That is not the protocol. The protocol is to clean, thoroughly clean, dry, inspect, package, and autoclave, heat sterilize. So cold sterilants do not play a role in that process. They're not recommended, and nothing has changed. N95 masks I'd already mentioned cannot be sterilized in an autoclave. Again, that's the same question. Okay, why do cassettes and instruments need to be dry prior to sterilization? If they're not dry, there's a couple of issues. If there are any water drops on it, then the sterilant may not penetrate because it can't penetrate water drops. The other issue is wet in, wet out. So the concern with wet packs coming out of the sterilizer is that they wick, which means microbes can get through the wet packaging. It's damp. So if you place wet instruments in the packaging, it's going to wet the packaging, and the drying cycle of the sterilizer may not be able to dry that package, which means it's subject to contamination as soon as it comes out again. There's a question on using N95 respirators versus ASTM 1, 2, and 3 during routine dental treatment. Routine dental treatment isn't indicated at the current time, only emergency, urgent care. So the answer is no, not only because it's not required for routine dental treatment, but because routine dental treatment shouldn't be being performed based on the recommendations. Do you think we'll need to wear face shields when we return to work? Many people actually already wear face shields because it helps prevent spray, splash, and spatter getting on typically a surgical face mask and their faces. The other reason people sometimes use a face shield is to protect against laser plume. So it is already an option. Again, will we have an updated PPE requirement? And I don't know. There's nothing to indicate mixing witch hazel into hand sanitizer. They should be used as they were formulated. They've been shown to be effective that way. So it's not going to eke out the sanitizer, and it may impact it. It's certainly not recommended for use. Again, prophies shouldn't be being performed at the current time. It would be a concern if an aerosol if they were, but that's not indicated currently. Have there been COVID infections originating from a dental office? I'm not aware of any COVID infection originating from a dental office. Okay. The prevents of hydrogen peroxide, is there a brand name that's most used? As far as I'm aware, there are 2 on the market that are 1.5%. One is Peroxyl, and I believe Listerine also has a 1.5% hydrogen peroxide. So if you go online and Google 1.5 hydrogen peroxide and either of those names, it may come up. Or Google the name and look at the ingredients. Can you cover an N95 respirator with a regular mask to minimize soiling and allow reuse? Yes, that would minimize soiling. You would hope if you're doing that, it would only reach the regular mask. Okay, there's been infection in dental office. From one person, I won't mention any names. It's meant to be anonymous. So I was talking about patients, healthcare transmission to patients at the time. I'm hoping you're feeling better. I'm sorry to hear that. Okay, and I believe that's it. I'd like to thank you. I hope this was helpful, and I'm sure we will see more updates and more information as we go forward. Like to wish everyone well, and stay safe and stay healthy. Thank you very much.